ठीक है, नो प्रॉब्लम, थैंक यू। So we are just waiting for 5 minutes, and then maybe a few of your friends are going to join, friends and colleagues, and then we are going to start with the last class on systems biology. Okay. So we are just waiting for 5 minutes, and then maybe a few of your friends are going to join, friends and colleagues, and then we are going to start with the last class on systems biology. Okay. So we are just waiting for 5 minutes, and then a few of your friends are going to join, friends and colleagues, and then we are going to start with the last class on systems biology. Okay. Okay, so about 8 or 9 people are over here. So let us slowly start, and then others can join in. And as and when we kind of people will start join the class. If you have any doubt, just put a message over here, and I'll try to get back to you. Okay, so today we are continuing with what we did yesterday. So today we are going to look it into a little more analytical way of the things of systems biology. So a couple of first slides are essentially the one which we summarized in the last two classes. So what is the learning needs for us is the basic things, that what is systems biology is. We need to define it, we need to understand it. And if you recollect that yesterday I had actually mentioned a very important thing, that systems biology is more of your approach, or more of your mindset of how you are going to address a problem rather than the problem itself. Okay. So most of you possibly might think that look, I'm not doing systems biology. I'm doing cancer biology. I'm doing immunology. I'm doing cell biology. So am I not able to do systems biology, or is it something a completely different thing? So don't think it is an alien science. So what is happening over here is that, what again and again I'm trying to tell you, is systems biology is actually an approach of how you address the problem rather than the a new discipline as such, right? It is just a way in which you kind of look into the data and plan out your experiments accordingly. And one of the most crucial thing in doing that judgment is to understand that there is an interactions between the system and the surrounding, right? The system is not in isolation; it is always interacting with the surrounding, and depending upon how it interacts with the surrounding, it will have a different effect. Number one, and number two is that biological systems, as I mentioned, that innumerable number of times, I just will keep on kind of repeating it again and again, that biological systems are inherently non-linear in its nature, and non-linear systems are the one where we cannot do a very good predictions, because of the reason that there is some kind of an inherent property of the system, because of which we don't have a direct correlation of the input and the output. So be aware of that facts, and therefore be aware of the variabilities which should come during the time of doing an experiments. The next point says about the standard analytical tools and for the large data sets. Remember, another very important thing about systems biology and compared to that of the rest of these other courses and curriculums is that in rest of the other things, in rest of the other courses, what we do is that we start an experiment, right? And then we carry on that experiment for some time, and eventually we get a good amount of data. And then once you get a good amount of data, then you actually start analyzing the data and then see that what does the data tell about doing the experiments, right? That is exactly the way in which we do a conventional biology. I mean, everyday biology, whatever we are actually doing. Systems biology is a little unique in the sense that here we start with a very large data set, and then we move in a backward direction and ask that, okay, this is the large data sets, what information I am going to make use out of it, what does this data set tells us. So the gross difference between systems biology and the rest of the other ways in which you are doing biology is that we don't do an experiment right away in systems biology. In systems biology, we already have a very large data set that could come from many experiments, like for instance, someone is doing a next generation sequencing, someone is doing mass spectrometry, someone is doing possibly a high throughput multi-channel flow cytometry, someone is doing confocal microscopy, someone is doing a histopathology and tissue arrays where they have hundreds of tissues and then kind of sorting all the tissues. So systems biology approach is starting from that data sets which we get from large number of experiments, and then doing the data mining out of it to see that what is the behavior of the data, what is the pattern hidden within the data. Does the data tells us any story, and what is that story? So it starts from a large data set rather than other sciences or other disciplines where we start with experiments and then produce the data and then that's it. That's the end of the experiment. Okay. So remember that difference. No, for that, in order to accomplish that, we need an advanced analytical tools. Like in the first class, if you remember, I told you that systems biology is actually an integration of computational biology, mathematics, physical sciences, of course biology, and a lot of statistics. So it is kind of a multi-dimensional thing, a multi-dimensional approach where we actually use so many different analytical tools to address our problem. So it definitely needs an advanced analytical tools. And needless to mention that computational biology is very important part of the systems biology, where you need to write codes and then do some fancy programming and then try to achieve the best thing out of your experiments and try to achieve the questions which you are actually been asking for. So these are all part and parcel of systems biology. And so that is something what is actually been discussed in this class. In the next class, in the next slide, what we are talking about is what is the motivations for doing a systems biology. So what is the motivations? The motivations are, so what is a systems biology approach? And again, I'm not going to repeat this because I just mentioned in the beginning of the class is that systems biology approach is actually something which you are going to articulate. You are going to train yourselves in doing it. We are going to take into consideration of the inherent nature of the systems, and then we are going to approach it in that way. So you don't need a separate kind of tools and techniques. Definitely you need to use those computational analytical tools, but then you don't have to train yourselves in a special way. All you need to do is that train yourselves in how you are addressing the problems and how you are actually going to tackle the problems. Okay. So the next thing is that why we need to take such an approach and how can one study systems properties. Now this is the one which you are going to give a couple of examples in today's class, and then you are going to cover those things. The first one, what is actually highlighted in the bottom panel is first look at the set of the gene expression data or genome expression data. I think if most of you may be aware of, if not, I'm just telling you that sometimes we need to find out that what is the change in the gene expression when you add a particular drug to a particular cells, or what is the change in the gene expression when a particular cell is starving, or what is the effect of a gene expression changes between a cancer cell and a normal cell. This kind of problems actually frequently we kind of address in day-to-day life, right? So the difference between a control and the test could be anything. So the test could be stimulation of a drug, chemotherapeutic agents, stop state versus effect states, some kind of a stressor versus non-stressors. Hundreds of problems are there where you could have control sets and you can have test sets, and then you get the data out of that. How do you know how to get the data out of that? One of the approaches is actually what we call as the genome expression data. When we say expression, we actually mean that we are talking about RNA expression over here. So we just try to find out that what is the change in the RNA profile, what is the change in the transcriptome, what is the change in the population of the RNA, what is the change in the transcriptional landscape of the cell between the control state and the test state. Or in other words, if we do these experiments, how exactly it is going to put in alterations and perturb the transcriptional landscape within the cell. In order to do that one, right, there are so many different ways to do that one. What is good about not systems biology per se, but good about this experimental science is that for the last 10 or 20 years we have something called the microarray, where people kind of put the RNA from say control samples and in test samples, and then find out that what is the difference in the expression and the populations of the RNA between these two sets, and from there they could conclude that what is the change in the gene expression. That's what is actually being mentioned over there. Of course, these days we have something called the next gen sequencing, where you can do a even more high throughput, even more advanced way, and find out the difference between a control state and a test state, drug treated and non drug treated. So these are the very fancy ways of doing this, because of the reason that there is a huge technological advancement, and because of that computational advancement along with that, together they kind of fueled this entire process, and now people can measure change in the gene expressions in a matter of just a couple of days. That's it. So it becomes very easy, very affordable. It's not very expensive, and it gives a lot of informations. If you do a microarray experiment, you're going to get information about 30 to 40,000 genes. Think about it, that if you're going to do them singly, one by one, doing an RT-PCR kind of an experiment, you're going to take years, right, to do or to integrate the expression changes of 30,000 genes. But in the case of microarray kind of a high throughput assays, you could do it just in one experiment in 24 hours, and next day you're going to have the data in your hands. So that's a huge advancement in this science. Okay, this is just an example of how the microarray looks like between the two different conditions. This is just an example. There's nothing serious too much about this. We are not even talking too much detail about the experiment. We're just talking, just to give you a flavor of exactly what is the measurement of the gene expression profiling. So this is two different conditions. Cells grown in condition number 1, cells grown into condition number 2. And then what we do is that we are asking a very simple and a very basic questions in a very naive way, that what is the change in the gene expression profile? How does the transcriptional landscape changes? How the transcriptome changes? How the RNA profile changes when the cells is in one condition versus in condition number 2? So the easiest way of doing is that cells growing in the condition number 1, isolate the RNA from them. Cells growing in condition number 2, isolate the RNA from them. Put it on a microarray slide over here. I don't have the slide right now. I should have brought it, but it's just like a glass slide. Just like an hemocytometer, which we actually kind of use to measure the count the number of cells. Just that this actually specialized slides with probes, which is already been sitting over there, and then eventually kind of label them with fluorescence dyes, and then take it under a fluorescence readers or something like that, and then eventually what we do is that we get and profile something like this, whatever is actually shown over here. So we'll have hundreds and thousands of dots, with the different colors. I'm just going to tell you in a minute what is this dots are. So just hold on with me. Possibly we will have a few slides where I'm going to tell you why something is green in color, why something is yellow in color, and why something is red in color. But this is just an overview, very simplistic overview. Of course, you can tell that this is kind of an summary slides where the microarray and how does it actually looks into. Okay, So I'm just uh, going to the next slide. Okay, this is something which we already discussed. So let's move on to the next slide. Okay, again, this slide I'm not going to spend much time. This is actually how the data output actually looks like when you put it or transfer it to an Microsoft Excel platform. Hold on one second. I'm just getting a phone call. Hello, hello. हाँ हाँ पार्किंग हाँ. अच्छा मैं ना लेकिन अभी एक मीटिंग में हूँ. मैं आधे घंटे 1 घंटे बाद ही फ्री हो जाऊंगा. ठीक है मैं देखता हूँ किसी को मैं भेजता हूँ. Is it audible today? Okay, so I just happened to look into my chat logs and I could see that Prachi has actually asked a question that what is the difference between emergent properties and extended phenotype? Wonderful question. So these are actually kind of the two sides of the same coin. So they are not very different from each other. So what happens is that when you have the emergent properties, that actually gives rise to an extended phenotype. So emergent property is the underlying principles or underlying things, the intricacies of that one, and the extended phenotype is actually the readout of that one. It's something like this. If you take a cell phone and then if you tear it apart and then look into it, you'll see a motherboard over there and lot of different CPUs and chips and over there. So those are the things which actually builds this entire cell phone, right? So because of that, the cell phone actually works as a one integrated unit. So when we talk about cell phone and its behavior, that's an extended phenotype. And what is the built-in within that cell phone, the chips and these microprocessors that gives a particular cell phone a 12 megapixel camera, someone with a 20 megapixel camera, some cell phone with an 20 hours battery life, some cell phones with 5 hours battery life. So these are the intricate detail within the cell phone, right? So those intricate detail within the cell phones are actually what we call as an emergent properties, because this actually happens as a result of complex interactions between the different components, and because of that integrated together externally, how it does it looks like, it looks like as an extended phenotype. Where And then why we say an extended phenotype, the reason being is that if you look into that phenotype, you cannot correlate directly with the genotype. As I've given you enough examples yesterday, that if you look into a genome of any particular organisms, nowadays we have over 500,000 organisms where the genome is already been sequenced and it's been actually lying in the public servers. You could always go and grab those genome sequences and do some fancy analysis out of it. And if you look into those genome sequences, none of the genome sequence tells that this person is going to be African, this person is going to be Australian, this person is going to be Asian and Asian Indians, and even in Asian Indians, this person is going to be living in Northeast, this person is going to live in Eastern region, this person has this food habits. Absolutely no, we cannot predict it just by looking into the genome sequence, because all what we see is a strings of A, T, G, and C. But then, how does the different phenotypes, even within a family, your siblings will have different phenotypes, right? How those finer detail comes into it's not just epigenetics, but it is also the emergent properties and extended phenotypes, because of which there is a complex interactions between the systems and surroundings, and we get in completely new sets of behavior. And the entire thing, emergent properties, extended phenotype, is a part of a very large universal set. If you think this as a subsets, this is a part of a very large universal set, and that very large universal set, which is actually part of it, is known as behavioromics, where you study behavior. not just phenotypes. We just don't talk about phenotypes anymore. We talk about behavioromics, where it is absolutely the behavior of an organism together, not just the phenotypes. So I think I could possibly answer your questions, if not completely, and if not very satisfactory, we can always talk about it, and in a later class, or whenever we kind of meet in the department or wherever it is. But so far, I think that much information is enough for us to know about the different things that between emergent properties and the extended phenotype. So, okay. With this, what I'm going to do is that I'm going to show you. So what is actually been right now on your screen is actually just an Microsoft Excel data sheet that tells you that if you do a microarray experiment, this is how the data looks like. Nothing more than that. This is some data. Don't bother about it. I mean, this is just I grabbed it from somewhere. Some yeast hybridization data for some yeasts. Growing into two different conditions. We have nothing to do with the biology over here. We're just going to show you, give you a flavor that how the data actually looks like if you import it into an Excel tool. And if you see on the top of it, if you could actually read over there, it's written as Matrix. What is Matrix? Matrix is actually the company that actually provides this service. One of the so many different companies that provides this service. Okay, there's an Agilent that provides this service. There's an Illumina that provides this service. So there are different platforms, and these platforms are actually finite details about when you're actually doing the experiments. Because if you are trying to do an experiment in an Affy platform, or I use an Affymetrix genome chips, then it is better to stick to that. You cannot use in one experiment an Affy chip, and in another experiment you cannot use an Illumina chips, because between the two different platforms there is a lot of confusions, and there is a lot of problems could be there. So stick to one particular platform, and then possibly you could have an uniformity in the data sets. So this is the finer detail which we need not have to bother right now in a theory class, possibly in a practical class that is going to be more relevant. Now the next slide is actually tell you that how exactly the data is actually being represented. Any microarray data is actually being represented in the form of what you call as the log of the fold change. You just not talk about fold change, you talk about log of fold change. What is the reason we do log? Because any one of you can actually chip in that what is the reason we put not a linear scale but a log scale. What's the advantage of doing a log scale? Okay. Okay, so the advantage of having a log scale, because we are not interacting one to one, so let me actually tell you the answer. The advantage of doing a log scale is that you can squeeze the scale. Say for instance, I give you an example. Say G expression of one of the gene has a value of, suppose, some fancy number, just give an example, is 1. The expression of the next gene is 20. The expression of the third gene is 200. The expression of the fourth gene is 5000. Now if I tell that this is the expression of 5 different genes, 1, 20, 200, 500, 5000, put it and show it in a graph. Now that will be a Herculean task, because you cannot very efficiently do an plotting when the data has been so badly scattered. Okay, because if you see 1 and then 20 and then 200, 500, 5000, if you look into the data set, they're all over the place, right? They're very nastily, they're scattered in a very nasty way. So it's very hard for you to kind of put them all together into one graph and tell us. If you wanted to show a 5000 expression change, then the one which has been showing an expression change of 5 is never going to show. It's going to be almost flat to the axis, because now the axis has been amplified million times. So in order to do away with that problems, in order to get rid of that problems, people, what they do is that they kind of squeeze the entire graph and entire axis by putting a log over there. So when you do a log over there, you kind of put them all and compress it. And this capacity of the log, or taking a logarithm, is just to do a compression of the data, so that you can kind of zoom in and look into everything together. It's something like if you blow up, suppose you're typing something in the Word file and you look it into a 200x zoom, then you'll see that just you are seeing 1 or 2 lines, right? But if you reduce the zoom to say 25x, you see the entire page over there. So that's the whole purpose of doing a log, is that you wanted to see the entire expression profile. So in order to achieve that, we squeeze the kind of axis, and one way of squeezing the axis is actually doing a logarithm over there. Okay, so that explains. So when you do a fold change, so most of the time when I was talking about expression things, we say that okay, control versus test, right? So we are curious, we ask this question is that how much time the test is different than the control. Suppose you have two cells and then you have added a drug into one of the cells, and you add in control or no drug in another cells, and then you are asking that okay, when I had added the drug, how much time the cell is proliferating more into that, in that drug added cells, how much time they're adding proliferating more as compared to that of the cells where there is no drug, or how much time the cell is actually being, how frequently the cell, or how much the cell is actually undergoing apoptosis or cell death in the flask where you've added the drug versus the one where there is no drug. So for that, what you do is a ratio of this fold change. So it is just a reciprocal of these two things, obviously, right? If something is x, something is y, in order to know the difference between these two, fold change is actually the ratio of x by y. So that exactly what you're doing is that we are measuring the expression. E actually stands for expression, and we are measuring the expression of the test, or the one of the test, which is actually the experiment versus the control divided by the control, right? And now we know for sure that when you do a log of division a by b, it's always log b a minus b, right? So that's exactly what is happening. So log of expression change of the test versus the expression change of the control is actually fold change, and this actually translates to log of expression of test minus expression of control, right? Now the lower panel actually tells you a very interesting thing is that if the expression of the control is more than that of the expression of the test, or in other words, if the test is less than the control, we say this is down regulation. You see over here negative sign, that's a down regulation, or this particular gene is eventually down regulated. Why? Because its expression is less than that of the control. See, expression of the test is less than that of the expression of the control. Now if that happens, then the value actually moves in the negative directions, because over here if you see expression of the test minus expression of the control, if this guy is actually smaller, then that entire thing actually becomes negative. It moves in the opposite directions in the negative axis, right? And we say that this is a down regulation. And on the other hand, the flip side of this one is that an expression of the test, if it is more than that of the control, then these overall things become actually positive, right? This is higher, this is lower, so you're subtracting a large number, a small number from a large number, so it is always in positive, and when it is positive, then it moves towards the positive direction of the x-axis, and we say this is an amplification or overexpression. So this is a down regulation of the gene expression. If the expression is less than the control, this is an overexpression of the gene expression. And if there is no change, if the test and expression of the test and expression of the control is exactly the same, then log of 0 minus 0 becomes exactly, there is no change of 1, and eventually we have that same thing, there is no change between the control and the test, and we say that no matter whether you have added the drug or not, it does not have any effect, because it is not changing the expression in the test samples where the drug is added versus where the drug is not been added. So that is the three possible scenarios: in positive change or overexpression, a negative change, and underexpression or down regulation, and no change, which is absolutely a scenario where we find that actually there is no change in the expression. Okay. Now this ratio of expression of this test by control is known as differential expression. So all we do is that we measure the differential expression. Many times we might have encountered the word that is known as DEG, which people actually use in short for differential gene expressions, right? Or differential expression of the genes, DEG or DGE, whatever it is. This actually shows the differential expression of the genes means we are doing the ratio of this test by control. Okay, very simple. Concept is very simple. Just once if you understand that, you are never going to forget it, because just very simple to all the other calculations which you actually do for the undergrads and other people. Okay. Now this is a graph which kind of a little busy graph, but again we are not going to spend too much of time, and we are going to see that okay, what is this graph? It's showing, this is actually showing you a heat map. Heat map is actually one of the ways in which you actually kind of represent your data. Okay. Now over here if you see that there is something called the log fold change over here, and this is a scale which is actually been placed next to the heat map, and over this the scale, the yellow color or the yellowish color or yellowish trend is an enhancement or upregulation or overexpression, and the bluish thing which runs in the negative scale is actually under expressions or down regulations or suppressions, right? And 0 is actually there is no change. Now suppose you did an experiment over here, 1 2 3 4 5. Suppose 5, sets of experiments is there, and if you do a gene expression values, if you have those values by fold change, where you measure the ratio for each of the genes, you measure the ratio by test, the ratio of the test by control, and then you got that value, and then you actually put it in heat mapper kind of a software, or you can actually do it into R, which is another computational tool, or MATLAB, or whatever it is, GraphPad, or whatever it is, whatever the comfort tool you are comfortable with, and do a heat map, then you see that this heat map actually shows this kind of a pattern. It's just an example. Again, now wherever you see a yellow color, it indicates that there is an over expressions or up regulations, and wherever there is a blue color, you can say this that's a down regulation. Suppose these are two pairs of the samples. You could see over here that these are the sets of the genes which is actually down regulated in this particular samples, whereas in this samples, corresponding samples, you see the same sets of the genes are upregulated, right? So that is how we actually read these particular heat maps, depending on what color code it has, whether it's an upregulation or down regulations. So heat map is actually a visual tool, and the visual tool actually gives you at a stretch, together, just in one consolidated way, exactly what is happening to the gene expression profiling. It looks from this one that majority of the genes are actually upregulated, because you could see that there is an predominance of this yellow color, right? But there is a less of the green color, sorry, your blue color, which shows there is less number of genes which is downregulated, but more fraction of the genes. It looks like almost like there is about 70-30 or 80-20 ratio, where more number of genes are actually upregulated and less number of genes are downregulated. Again, this is a very selective example, so don't take it as an universal. This is just one thing which is actually picked up from somewhere. Okay, this is the one which I just wanted to show, and again we are going to move fast. Okay, let's see. Any other queries? Anything? So far, whatever we discussed, we are slowly navigating into a little analytical thing. So before that, just to make sure that we are actually all in the same page, and no one of you is kind of still scratching your head to find out what we actually discussed. Is there anything? Just put in the chat window. This is right in front of me, and I can re-explain to you. I can go back to the slides, and we can talk about that again very quickly. Okay. So let's to come. If you have a doubt, just feel free to put it in the chat window. I am always looking at it, and then I'll kind of catch hold of the question. Okay, this is what is known as the Rosetta data sheets. What is the Rosetta data? Well, this is something which we don't have to break your head. This is just how a microarray extrudes. We're all talking about microarray. Remember, right from the slide number 2, we are all talking about microarray. And what is microarray? It's an experiment to measure the change in the gene expression, or to find out the differential gene expression, or to find out how the RNA profile changes, how the transcriptome changes. That's what is expression array is actually doing it. The name itself suggests. So when you do an expression array, then what happens? I think we should have a slide to show you the raw data actually looks like. So this heat map, what I showed in the previous slides, is actually very sophisticated filtration out of that, and then it's a very sophisticated of representing the entire data. But actually, if you go into the instrument and actually look into the instrument, then the data actually looks like something like this. This is another Rosetta data or Rosetta plot, where it's kind of dots and points here and there. So all you need to do by doing some smart algorithms and writing some smart computational tools is that convert all of this, this uh, dots here and there into something which is more meaningful and more easily interpretable. So you could interpret it very easily and then actually understand what's happening, rather than representations in these forms. But this is actually what is happening in the core of the instrument, and therefore I kind of bring the slides to tell you, to give you the feeling that this is exactly what is happening over here. Okay. Okay, now next of the slide is actually one of the experiments which I'm going to tell you. It's a very small experiment, and then we are going to spend more time into the data which actually comes out from the experiments. Okay, so what is this? This is actually a plot. This is actually a plot of the distributions and fold change. And over here, what it actually shows is that there is a yeast is there. Okay, one yeast is there, and someone actually deleted a gene that is known as ADE. So this is an ADE deleted yeast. So there is yeast, you know, so one of the yeast samples, so one of the yeast population has ADE deletions, and the other yeast population does not have the deletions. And we are asking a very basic fundamental question is that, okay, is there a change in the gene expression profile if this AD1 gene has been deleted? Or in other words, let's compare the gene expression profile between a deleted population where ADE1 is deleted or knocked out versus the population where it is not deleted, that is the control populations. Okay, so someone did a microarray experiment out of it and they've got a graph like this. This graph, if you see, is very narrow, very tight, very congested, and very focused at one particular point, right? Just by looking into the graph, we could find out that very easily it is, you could always interpret that this graph does not have wings in this direction, does not have the waves moving in this directions. Or in other words, what you could see is that majority of the data sets are at the zero axis or the 0 point. When I mentioned in the previous thing, when the zero is there because of the fact that there is no change in the gene expression, right? Because that's why how the log 4 change is going to be zero, because it is expression of test minus control. Test is same like control, then test minus control is 0 minus 0. You're going to get a value of 0, right? So that is exactly what is happening. So quickly, by looking into this histogram, we could actually conclude that whenever there is an ADE1 deletion, then the global transcriptomic profile does not changes too much. So that's the interpretation very quickly, because everything is very tight, congested, focused, and not spreading in the axis, which shows that the differential gene expression is not there. Differential had there have been a differential gene expression, you would have graph spreaded right over this particular regions, left hand side, right hand sides. But we see that everything has been concentrated at just one point, and that is zero, and which shows that no matter whether you delete it, no matter whether you knock it down, this particular gene, it does not have much influence on the overall gene expression profile. Or in other words, in an ADE deleted yeast, the gene expression profile is almost equivalent than to the one population where it is not deleted. So that is the interpretation from this graph. Okay. Now I'm going to remove this Rosetta one and again actually show you, pull up another graph. Okay. These are the two different interpretations of two different kind of data sets. Okay. This is another deletion. This is an SSN6 deletion in the case of yeast. Okay. Now, if you see this graph and if you compare that, and if you remember that, recollected what we talked about the ADE1 deletions, you could see that the topology of these two graphs, the geometry of these two graphs is very different. That graph is more or less always concentrated. The previous one right over here, right at the 0 point, very narrow, no spreading, no wings here and there. But in this graph, you could see that the entire thing has not only been concentrated over here, but it's also been extended into this side. Or in other words, we could actually then know what are these two? These two are actually the outliers of this one. So we are not talking about them. This is where any data, if it is in more than this, then you are going to say that okay, this data set is outlier. We rather discard it, or we rather not put it into these particular calculations and going to see it separately. So what I had actually mentioned over here is that, so from this one, what we could interpret is that compared to ADE1 deletion, which does not have a profound influence on the gene expression profiling, if you do an SSN6 deletion, then there is so much change in the gene expression that you get a spread like this way, right? Or in other words, there is a lot of differential gene expression. So that's what exactly has been said, that SSN6 deletion mutant exhibits large differential gene expression, because it is not being just concentrated near the zero, but it actually is stressed in both of these two directions. So this shows that SSN6 and ADE, they behave in a different way. Depending upon which one you are going to knock it down, we will have a different influence on the gene expressions. And this also tells us a very interesting thing is that SSN6 is possibly a very central hub of a transcriptional network. And why? Because it is actually been controlling the expression of so many different genes. See, once we remove SSN6, so many genes expression changes has taken place. Whereas ADE1, if you remember, whether you removed it or not removed it, there is not much population of the genes whose expression changes. And therefore, this makes us to think that SSN6 possibly is actually a very important master regulator because its absence has a profound influence on the transcriptional landscapes and the transcriptional profile of this entire experiment, right? That is how we are going to interpret it. Okay, this is the next one that actually tell you a small calculation which is actually been done by just doing some mean and then standard deviation out of it. So not much to talk about. Most of you are possibly aware of this one, so we are just going to go through this slide rather very quick. So mean and the standard deviations of these distributions. Okay. So standard deviations means how much each of the values are actually been deviated from the mean. So that is exactly what you are actually been trying to do in this case. Okay. So see the formula of the standard deviation. It is the values xi's and how much they have been scattered, how much they are different, how much they are moved away from the mean, and then you do a square root of this one, and then that's how we get on standard deviations. Okay. This is the outliers which I actually just mentioned in the previous discussions when I was talking about the previous slides. These are the two outliers which is actually sitting at the corner of the graph. Okay. Okay. Let the outlier to come, and then eventually we will move from the slides. Okay, that's the outlier. Okay. Now, if you look into this distribution between these two knockouts, and the two profiles are now side by side, the ADE1 deletions and the SSN1 deletions. And if you could see from here, from these distributions, you could very clearly see that when you have an ADE1 deletions, then the standard deviation values are rather very slow, very less, right? Because everything is very tight. So all of them are all concentrated near the mean. On the other hand, if you have SSN6 or SSN1 deletions, then there is a big scattering. And as a result of this, the standard deviation is also very substantial. Now, standard deviations become substantial whenever we haven't changed or alterations in the gene expression profiling. So, the value of the standard deviation is also another indicator which actually tell you the spread of the data, how much the data is scattered and how much the data is spread, which could be because of some technical reasons or it could be also because of some biological reason. Because if you remove the gene, then there is a profound influence on the expression of rest of the other genes, and therefore the distribution becomes very wide. And now the distribution becomes very wide. It will always differ from the mean, and therefore we will have standard deviation which has been popping in from nowhere, because the standard deviation happens only when there is a deviation in the mean. And deviation from the mean happens only when there is a scatter in the data, whenever there is a distribution of the data in and spread of the data. And that actually happens only when the log two-fold change is being substantial in the control and the test. So that actually gives you again an idea that what kind of things is actually been happening. Okay. I'm just pulling up these very important slides and going from there. The next important thing what we will discuss, of course, it's not the end of it, but we are going to discuss this one. The next important thing is what is called, so in between, we have this, students t-test and all these kind of things, which I just kind of, avoided it, because this is something either we are going to study and we have possibly already studied it. So we are not going to, doing too much on that one. Okay. Now, okay. So this is something a little important, which is known as the Pearson's correlation coefficient. What is that Pearson's correlation coefficient? डॉक्टर साहब मैं एक क्लास में हूँ, मैं अभी आपको निकल के मैं आपको फोन कॉल बैक करूँगा, मे बी इन द नेक्स्ट हाफ एन आवर. ओके. Okay, so what is this Pearson's correlation coefficient? Well, this is a kind of an correlation coefficient which gives you an idea about how the two data sets are actually being associated with each other. What is the relationship between these two data sets and how they are related with each other. Now, it is a measure of the linear correlation or dependence between these two variables x and y. Okay, Now, Pearson's correlation coefficient is something which we extensively use it in a large data sets, and it has three possible values. Any value of the Pearson's correlation coefficient values can range from -1 to +1 with a value of equal to 0. So this is the way in which the data can be interpreted. If you look into the graph, so when you have a Pearson's correlation coefficient equal to -1, this means the two components or the two data sets are anti-correlated. What does that anti-correlated means? Means if one data set increases exactly at the time, the second data set actually reduces. Or if in two data sets, the gene expression in one of the data set increases, the gene expression in the other data set actually gets reduced. So that actually happens only when you have the Pearson's correlation coefficient equal to -1. Pearson's correlation coefficient is going to be U +1 when the change in one gene expression is also associated with the change in an equal amount of the next gene expression, other gene expression. And therefore, if you have two different data sets, and if you see that in this particular data set, this gene expression actually increases, and exactly in the other data sets, the same gene is also being increased, then you say that, or same or the different gene is actually increased, then you say that, okay, these two genes are positively correlated with each other, and then the Pearson's correlation coefficient equal to +1. And we have a scenario where there is no correlation at all, or in other words, if one gene expression changes, it doesn't matter, the other can increase or decrease, anything can happen, and then we say that there are no correlation at all, and that gives the Pearson's correlation coefficient value equal to 0. So that is all the three possible values of Pearson's correlation coefficient. A positive +1 values means completely correlated, positively correlated. A Pearson's correlation coefficient equal to -1 means completely anti-correlated. When one increases, the other decreases, and the Pearson's correlation coefficient equal to 0 means they are not at all correlated at all, or there is no relationship between these two data sets. Okay. That's something what actually we discussed in the slides. And remember that between this value of -1, 0, and +1, you could have numerous number of values, right? You could have value of 0.38, you could have value of 0.73. So depending on what kind of value is there, you will have a different interpretations of this Pearson's correlation coefficient. Okay. So again, this is something which we have to do it to get a more hold of this one. Okay. This is just a graph, just a cartoon that actually tells you that depending upon the different values of this Pearson's correlation coefficient, you could have the scatter, pointed in different directions. Whether it's been inclined, ideally, if you see a perfect positive correlation, it runs through the 45-degree diagonal axis. But if it is not, then it's actually distributed in a very random way. And we have all the different possible distributions, but all of them must have a value within the range of -1 to +1. Okay, that's something which is very important. We are not going to discuss, we are not going to talk too much about this, or not going to get ourselves confused into this. Okay, this is the one that actually tells you from the heat map what are the two scenarios of positive correlation and negative correlation. If you see over here, these are the two different conditions, or these are the conditions under which we are measuring the expression of two different genes, gene 1 and gene 2. So this is what is the correlation in the expression profiles of gene 1 and gene 2 in a particular data sets. Now if you could see over here, wherever there is yellow, suppose yellow is down regulation, and suppose yellow is up regulation and the blue is down regulation. So wherever there is an yellow in one of the sets, there is also a yellow in the next sets. Wherever there is blue in one set, there is also blue in other sets. Wherever there is yellow in one set, there is also yellow in another sets. Or in other words, when you say that these two genes are behaving as if they are actually being joined together, right? As a co-joined genes. Because if one increases, the other also increases. If one decreases, the other also decreases. And this is an example of a positive correlation. And R1 is a relationship or correlationship between gene sets 1 and gene set 2, and its value is almost equal to 1. Because whenever we have this particular thing happening, that one is actually going up, there is also the place where the other one is actually going up. And whenever one is going down, the other is actually also going down, telling that these are positively correlated. But if you look into the lower panel, you could see that there is actually absolutely no correlation. Wherever there is yellow, there is sometimes blue. Wherever there is blue, sometimes there is gray in color, or no change in the expressions. You could see there is not following any pattern. It's not that one is going up, the other is going up or going down. So it is very random, very chaotic, and therefore we say that this does not have a very good correlation. And then let the correlation be 0. And 0 correlation means these are completely independent. One does not have the effect on the other one. And that is something which is very important to see that what is the value of the correlation coefficient and how we are going to actually interpret out of it. Okay. Now over here in the third example, if you see, wherever one is going up, the other is going down. In the first one, wherever one is going up, the other is also going up. Where one is going down, the other is going down. But in the third example, you could see that when one is going up, the other is actually going down, and so forth. And this is actually possible only when there is an anti-correlated with each other, with an Pearson's value equal to -1. -1 indicates that they are actually moving in an opposite direction. Whenever one is going up, the other is going down. Whenever one is going down, the other is going up. So it's kind of an anti-correlation. This is the different possible topology of this Pearson's correlation coefficient. And the first one is positively correlated because this is moving in the positive direction of the x-axis. The value is equal to 0.8. Not bad, pretty good correlation. And the second one, if you see over here, they also the value is equal to 0.8, but then kind of it's not in a straight line like the one which is actually shown in the first figure. That is actually a little more curved. So even though the correlation coefficient has some value, don't take it as something which is very universal. You need to interpret this data individually, case by case basis, and try to reach to a conclusion. That is very important. Okay, just think about it. Okay. Now in this case, if you see the correlation coefficient, the value is equal to 0.8, which shows almost like 80% correlation, positively correlated because the value is equal to positive, right? Over here, if you see that this one also has an 80% correlation, even this one has 80% correlation, this is also 80% correlation. So there are key difference between these two, that this is not only correlated, but this is also collinear. That is very important. That is not just correlation, but it's also collinear. And the second one, third one, fourth ones are actually correlation, but there is not collinear, right? This one is still a little better, but the rest of these two are not have a correlation but does not have a collinearity. Now when we have this kind of scenario where there is a correlation but no collinearity, then we actually end up in having a different sets of conclusions, and that is again very important. And then we say that even though there is a correlation between these two, at high correlation, it cannot be cause and effect, right? Because of the reason that there is a non-linearity in this one. But if you see in the first one, there is a linearity. There is a linearity in this one, and this linearity is because of this fact that each one of them can actually has a dependence on the others, right? So they follow a pattern, and therefore we can say that there is a cause and effect kind of a pattern is over here, but we don't have a cause and effect kind of pattern in any one of the rest three. So what this slide kind of tell you is that if you just have a correlation coefficient value of anything, 0.8, 0.9, 0.1, 0.01, 0.001, if we have a correlation coefficient, don't just jump into conclusions without looking into the collinearity of it, because you could still have correlation coefficient of 0.8, which is extremely high or very good in three different scenarios where it is not even collinear. So only the first one is collinear, but the rest of them are not collinear, and therefore even with having a high correlation coefficient values, they really are not very crucial or very important because of the fact that they are not collinear with each other. Okay, now I'm going to bring an example, and this example is going to help us in kind of reaching to what I'm actually trying to tell you. Okay. This is an example of something called the HRT, or hormone replacement therapy, which is proved to be very beneficial in CHD, which is known as congestive heart disease. And over here, if you see that we have a high value of correlation coefficient, or in other words, those females who after menopause undergo what he calls in hormone replacement therapy, those females are actually supposed to have a good protection from congestive heart disease. अच्छा कुछ classes है क्या? They will have a good protection against congestive heart disease. And this is the idea that was actually floating back in 80s, in 90s, in early 2000s, that any female who has postmenopausal, any female who has a hormone replacement therapy, they do pretty good in coronary heart disease and cardiovascular disease, because of the fact that hormone replacement therapy is possibly helping it. It's beneficial, right? Now, even the hormone replacement therapy is actually beneficial. Can we conclude that, okay, these are correlated with each other? In other words, if you have hormone replacement therapy, then we find that is actually been associated with betterment of the life, and there is less of coronary heart disease. But can you say that just because of the reason that there is a hormone replacement therapy, there is a protection against coronary heart disease? Or is it that anything could be there? So people were actually scratching their head initially to some kind of a thing where just by seeing the correlation coefficient, they kind of reached the conclusion. But over now, we actually became more analytical, and we just not look into the correlation coefficient, but also into the entire picture together, seeing how and where, how much they actually overlap with each other. So, okay. It is actually been found that the females who actually undergo hormone replacement therapy, okay, HRT is a pretty expensive procedure in United States and UK. So those females who actually undergo hormone replacement therapies, they are mostly the persons who actually belong from an affluent society, right? So I'm just telling you the story why it actually becomes very strongly correlated, because those females who actually undergone in hormone replacement therapy, most of them are actually from an extremely good family background, economically very sound and economically very affordable. Okay, they could afford this particular procedure, and then they had definitely a lower risk of cardiovascular disease. But is that the cause? We don't know. Well, if you dig into further, then we find that those females who also undergo hormone replacement therapies and who are possibly belonging from a pretty well-off family, economically very stable, these are the females who are also been aware of these health issues, and therefore they are actually more prone for taking care of their health. They are more prone for taking good vegetables and fruits and doing exercises and all the precautions. But those females who have hormone replacement therapy, for those females where hormone replacement therapy is good, we can say or we cannot say that it is because of this, there is a protection against cardiovascular disease, because of the fact that those females are also the one who are actually taking care of their health more than those females who are from a poor economic backgrounds. Now I'll give you an example, just this example, and I'm just trying to make you a little more clear that just doing a hormone replacement therapy and concluding on that this is going to be protective against cardiovascular disease or coronary heart disease is possible, and over stretch of the conclusion it's not true. And the reason why it's not true, because those females who have actually undergo hormone replacement therapy can also afford some better lifestyle alterations like morning walks, yoga, and then affording good foods and everything together. And therefore, it is very hard to piece together that is it really the hormone replacement therapy which has given the protection, or is it because of the reason that they are actually been going through a very nutritious foods and couple of exercises and every kind of and enough rest, and therefore that has actually provided and benefit to the CHD. Whereas if you take the same HRTs for a female who is belonging from weaker socioeconomic groups, either way she is not going to afford for these expensive procedures. Not only that, and even if someone actually volunteers that kind of do it, but at the same time someone pays for that, at the same time it is also very feasible that those females will not have an access to good food, good nutrition, a good house to stay, and a society where there is less of infections and other kind of problems, right? Any kind of precipitating factors, and therefore those females are not going to do good. So they are not. So any hormone replacement therapy is going to do good, not because personally because of the procedure, but because of the so many other things that actually helps to reduce the risk. So even though we have a very high correlation coefficient, we really don't have a very good collinearity. In other words, you cannot say that is the reason for these causations. So high correlation or high Pearson's correlation or does not necessarily mean that we are going to have a causation kind of an effect or causal effects, right? It could be so many different things that could actually make it a little more difficult to interpret having a direct relationship between these two. The other example is actually p53 mutations in cancers. Now, if you take p53 mutations, most commonly occurring mutations in cancers, and if you see over here in many of the studies, the people who have p53 mutations has cancers, but then they have variabilities in the cancers. In other words, it's not that they had all the signs and symptoms which is same, but it's actually spread over an entire large sector. What is the reason for that? The reason is again p53 mutations. Okay, it does actually offers the cell a different phenotype, but at the same time, if you see over here, the p53 is actually the center of the hub of interactions with so many different vital proteins. Now, if the p53 has been gone, or if the p53 has been mutated or not functional, okay, there are other proteins which has been actually there over close to this p53, right? They all work together as one common network or one common kind of an landscape together. And this is what is actually important, because it is not just the P53 that decides the fate of all the things that is happening within the cell, but it is the P53 and its partners which actually together comes, and takes care of the phenotype of the cells. And that is actually more important rather than just alone the P53. So that is what we are actually trying to understand this, because most of you will be aware of the P53 actually undergoes what you call as an MDM2 mediated degradation. So these are the factors which regulate the level of P53 dramatically. Okay, and that is where we actually found that P53 is actually the center of the hub of many interactions, and its cells can be regulated by multiple different ways. And unless until we understand them and actually try to interpret from those findings, it is very hard for us to do just one experiment and conclude on P53 that this is always predictive, or this is actually been doing this kind of functions, unless and until we actually do in a systems biology approach whatever I am actually showing you over here, and actually reach that particular conclusions. So the next thing, so assuming that we had actually understood this, okay, so the next thing, so bear with me a couple of slides because I don't want to drag it further. I'm just going to finish it up today itself. So this is another slide that actually shows you what you call as a hierarchical clustering. What is hierarchical clustering? Well, this is one way of taking the data and then actually looking into it, that whether this data actually has the components which could actually be joined together as one family. That is what is known as hierarchical clustering, or very important type of clustering of the data. Now, before even going into the clustering of the data, I'm just going to put in word that what exactly do we understand by clustering of the data. So if you do an say microarray experiment and mass spectrometry experiment, then you have a very large data set that is delivered to you, right? Hundreds and thousands of pages sometimes. Now what are you going to do with these large data sets? Well, we are going to take these large data sets and going to find out what are the pattern which is actually hidden in the large data sets. In other words, just by having that particular data set is of no good use unless you kind of do a data mining on the data sets. And one of the data sets, one of the way of doing the data mining is to first try to find out how many different variabilities or what are the different heterogeneity within this data. Okay, and this is actually going to explain with these examples, and I'm going to give you real life examples where possibly it's going to be even more easier for us to understand. Think about it. So in the case of an hierarchical clustering, what we are trying to do is that we are trying to link members in terms of the similarity in the functions. Link members in the terms of the similarity of the functions. Like for instance, let me give an example. Suppose we take 3-4 different medical colleges, say AIMS, PGI Chandigarh, and then Safdarjung, and then say for instance Japan Medical College. Okay, these are the 4 different medical colleges, institutions, academic setups where we kind of teach the people which is undergrads, and then actually we are trying to find out what is the correlation coefficient between these two. Okay, or how do we interpret that data? Okay. Now think about it. AIMS, okay. What is the relationship? We have these 4 different medical colleges, AIMS, Safdarjung, Jipmer, and say for instance other hospital is there. Another one hospital is there, say MAMC is there. Okay. Now what we do is that we know for sure, if someone says that okay, this is the data components of the data, how can you find a pattern within it? Well, the way of finding a pattern within this data set is looking to individual values with the help of computational tools and trying to draw a tree out of it. What is the tree? Tree is a kind of thing which is known as dendrograms. Okay, that I'm going to show you in the next slide itself, and the dendrogram actually gives you a fairly good idea that is what is actually been happening in which particular regions. Coming back to an example, suppose if you have these 4 different medical colleges, now we are quickly trying to find out okay, what is the relationship between them. Okay, one of the relationship between them is that AIMS has an MBBS program. Okay. And even Subhradip has an MBBS program, right? But for that matter, even Jipmer has an MBBS program. So what is the difference between them two? Okay. Now you put a second condition of MBBS programs are very proximal to AIMS. Well, okay. Once you put this condition, then you will have only one medical college which usually comes in, and then Subhradip Medical College. Because not only it has also an MBBS training program, but at the same time it's also very proximal to AIMS. And as a result of these 2 different facts, okay, if you have 4 different medical colleges, quickly you will see that we are going to try to put AIMS close to that of the Safdarjung Hospital. Or in other words, you could also say that Safdarjung Hospital can be clustered together with AIMS because of the reason. Because the reason is that their behavior or their way in which they operate is very similar. AIMS has an MBBS program, that hospital has Ames has Ames and Safdarjung is very close and proximal to each other, and therefore we put them as one cluster, right? Now, what are we going to do that? We are going to slowly expand the cluster and then see that who else can be related to this particular cluster. Okay. Now think about it. Safdarjung Hospital and Ames. Okay. And now we still have two more members which are not clustered together. That is Jipal Medical College and another one is the MAMC, right? Now we ask this question, okay, say for instance, which are the two hospitals which are actually being operated by the central government and which are the hospitals which are autonomous, right? Now, the moment we put this second condition, you could see that from this entire data set, only subdivision hospitals and possibly, we have subdivision hospitals and the MAMC, which will actually come as, the government operated hospitals, right? They are not autonomous bodies, they are government operated hospitals. Okay. So now that these two behaves together, we are going to put a club together the Subdhajing Hospital with the MAMC. So previously you see that we had clubbed together Ames and Subdhajing Hospital. Now by adding another condition, we are clubbing Subdhajing Hospital with MAMC. And what is that point? By which we are actually doing these clusters? Because in the first one you ask that, okay, who has the teaching MBBS program, who are proximal to Ames? Okay, Subdhajing Hospital popped up. So we correlated that with Ames and seal them together. The next important thing what I'm trying to do is, asking the question is that how many of these are actually operated from the government setup. So we find another two of them and we kind of correlate this. Hello? और गाड़ी में बैठ सकता है हाँ क्यों सब कुछ नहीं ये नो अच्छा गाड़ी अच्छे ठीक है अच्छा हम चाबी दे पाते हैं अच्छा ठीक है ना Okay, so with the help of this clustering, what we are slowly doing, what we are slowly doing is that we are trying to club together the components which are similar kind of a behavior. Okay, and slowly we are going to grow the tree in large numbers, and that is exactly what is known as the hierarchical clustering. Very quickly, so hierarchical clustering is the one in which we try to correlate the components which are being joined together or linked together and then try to put them into the similar bins. As I mentioned that if A is related to B and if B is related to C, then we say that okay, A, B, C actually forms one clusters. Need not necessarily that A has a relationship with C, but since because of the reason that A is related to B and B is related to C, so we can put A, B and C into one bin, into one basket and say that okay, these are clustered together. Now suppose we find that now B is actually related to F also. Then we can say okay, that okay, A, B, C and F are all joined together as one clusters. So slowly the cluster grows in size because of the reason that we are actually finding out the relationship between the different components and therefore we eventually build up a large dendrogram kind of a stuff. Okay. Now we had reached the end of today. Actually, I had to also push up because there is some kind of class which is going to start. But then I thought of finishing off this now. Um, is it possible for you to be there tomorrow?